This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 59, recorded on August 30th, 2013. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
Back from Australia.
Down under. Back from down under.
How was it?
It was fabulous. Fabulous.
And were there parasites?
Didn't notice.
That's the same answer you gave last time when I asked you. I didn't notice.
I wasn't down there. I wasn't down there to notice parasites. I was down there to participate in a meeting called the Australian Leadership Conference.
Why were you there at a leadership meeting?
That's a very good question, and I actually still haven't figured that out, but it doesn't matter. They paid for the trip, so I was happy to go. In fact, they paid for my wife also, which was even better.
Wow.
So we both went. And
nice. How long does it take to fly there?
From where?
From here, New York City.
From New York City, it's a 6-hour flight to Los Angeles.
You didn't get a direct flight?
Then it's a 14-hour flight. You can't get a direct flight.
No?
No, they all leave out of the West Coast.
Oh, too bad.
So you either leave out of San Francisco, Seattle, or Los Angeles.
Because I'm going next summer, so
yeah, I know.
That's why I'd like to know.
I think you should take your kids to Disney World in Los Angeles for a week and then go to Australia.
No.
Well, the flight over was 14 hours.
It's been suggested that I stop over in Hawaii.
That's a little bit out of your way.
It's a nice place, though.
It's gorgeous.
I was there.
Gorgeous.
Anyway, my plan next summer, July, Australia. I'm going to a meeting in Melbourne. And then...
It's not Melbourne, it's Melbourne.
Melbourne?
Melbourne.
All right. Anyway,
Please.
I'll do a podcast at the meeting. And then I would like to go to one or two other places where people would like to have a TWiP recorded.
That's true.
And do a TWiP with local virologists.
Yep. So University of Melbourne is right there.
University of Melbourne.
Is right there.
Melbourne.
Okay. And then the Walter and Eliza Hall Institute for Medical Research is right there as well. And they have lots of virologists there. And it's a wonderful place. I spent my sabbatical there in 1986.
Well, I have just less than a year to plan my trip.
You do.
So hopefully I can. What do you think about that?
I think that's a lot of time. But I'll be happy to advise you any way I can, Vince, because we spent some wonderful quality time before the meeting in places like Darwin and Kakadu National Park and Alice Springs and Ayers Rock and the Olgas and Kings Canyon and finally into Sydney and up into Hayman Island, which is where the meeting was held, which is way above Brisbane, as they pronounce it, not Brisbane, Brisbane. Brisbane. They just they sort of cut short their last syllables. So I'm back.
Welcome back.
Raring to go.
It's nice to have you back because I like to engage you for TWiPs.
We have lots of stuff to talk about. Today, we should probably call it today potpourri, because we're going to discuss little snippets and bits from various subjects. It's not going to
be potpourri.
Okay, fine. We'll
think
of something.
You can call it whatever you want, but
yeah,
I have a title, Avian Malaria.
Yeah, but we'll talk about that. We'll talk about some other things, too, though.
No.
We have to.
Do you mean like cyclosporiasis?
Cyclosporiasis.
Why can't I remember how
I
to
don't
say
know.
that?
It's a cycle. Just remember cycle.
Cyclosporiasis.
Cyclospora chiatenensis.
The last time we recorded, it had just expanded.
Yeah.
But the source
of
the infection, or at least some of the infections, hadn't been determined. And then
true.
You left for Australia. We couldn't update. See, this is what happens when you travel.
But I'm back. We're updating. Here we are. We're doing it. We're doing it.
It's over 2 weeks.
So there's a company called the Taylor Farms, which I find highly not amusing necessarily, but it sounds like they don't farm anything. Rather than they package products delivered to them from other places, because the Taylor Farms.
Taylor Farms is an American company.
It is. It's in Colorado, and it's the company that could be traced back tooth by the Centers for Disease Control, who then revealed that their products are being packaged in northern Mexico.
So
they're leafy green products, and they're then being delivered en masse to a variety of outlet stores, particularly restaurants.
Right. So as of today, there's an update on the CDC website. We'll put a link to this.
Good.
There have been 616 illnesses in 22 states.
Wow.
Arkansas, California, Connecticut, Florida, Georgia, Illinois, Iowa, Kansas, Louisiana, Minnesota, Missouri, Nebraska, New Jersey, New Hampshire, New York, including New York City. Remember I asked you last time?
You did.
Ohio, South Dakota, Tennessee, Texas, Virginia, Wisconsin, and Wyoming.
But the epicenter was Iowa and Nebraska.
Most of the illnesses were from mid-June to mid-July.
Yeah.
8% were hospitalized, no deaths.
Right.
So
it doesn't kill you.
In Iowa and Nebraska, so the first time we talked about it, which was 2 episodes ago, that would be 57.
Right.
We went through the whole biology of the cyclosporiasis. I still can't get it.
That's good. You're going to get it.
Anyway, health officials in Iowa and Nebraska have concluded that restaurant-associated cases were linked to salad mix produced by Taylor Farms de Mexico.
Right.
And then on August 12th, the company has suspended production and shipment of any salad mix, leafy green, or salad mix components from its operations in Mexico to the U.S. On August 25th, with FDA concurrence, the company resumed production and shipment.
Really?
So Dickson, they grow the leafy greens in Mexico. They package them there, or do they ship them bulk to the U.S. and then package them here?
It's a great question. No, they package them in bulk in Mexico, and then they're shipped someplace for distribution.
All right, so they're contaminated in Mexico then.
That's correct.
By a worker presumably shedding, what's the name of the thing they shed?
The oocyst.
Oocyst, the thing. Do you like that scientific
term?
I
love it, Vince. I love it. It's very virological of you.
And then it gets packaged, and those oocysts are fine in the bag and through the shipment and all that. They don't.
Not only that, they have to incubate outside the body for at least 3 or 5 days before they're infectious.
Remember perfect.
And that's what screwed up the epidemiology investigation because there's this delay. It's not an immediate thing. So you can be 2 or 3 days down the road someplace else from the source, and then you get a disease. Then you can transmit. So the packages have to sit around for 2 to 3 days too, Vince. That also tells you that it's not a very fresh package of salad.
So this is a effect of global food production and distribution,
always, foodborne illnesses of this sort always crop up.
Is that no pun intended?
It was not a good choice of word. I'll rephrase that. They always pop up unexpectedly because the inspection system is not accurate enough or vigilant enough to catch every single outbreak before it occurs. There are spot checks, and these spot checks are not adequate enough to catch these things. And most of them are not considered serious illnesses, including this one. This is a protracted diarrheal disease. It doesn't kill anybody. It makes you sick for maximum about 50 days of diarrhea, which is a very, very uncomfortable situation. But for most people living in the tropics, that's normal. So you get a little taste of what it's like to live in the tropics for a while. Mm-hmm. And then after that, you're all better. Okay? I don't know of any connection between cyclospora and immune deficiencies, so I really can't speak to that issue, although there may be some connections. And maybe this outbreak would point that up.
Now, in Texas,
yes,
the cases in Texas are not connected, and there have been a lot in Texas.
That's right.
They have not connected to Taylor Farms produce.
Exactly right. So there are several outbreaks.
Isn't this weird that you would have 2 different sources?
No, not weird at all. It's like when I was a diagnostic technician for the hospital, we were taught that just because you've found something under the microscope from a patient's sample of stool or blood or sputum, it doesn't mean that they don't have something else also. So you have to keep looking until you've done the whole sample.
I'm showing you an epi curve for the different states.
And I see 2 epidemics there.
So Texas has a different curve from...
They do.
What's the other? I can't see
Iowa.
it.
That's Iowa. The Iowa curve. Yeah, that's right.
So different sources.
2 sources.
But they're happening in the summer. Is that just because that's when we're eating lettuce? No, we eat lettuce all year round.
We do.
So why is it happening in the summer?
I have no explanation for that.
It's just coincidence.
It could be the same worker, by the way.
But they're not,
Coming up from Texas or from Mexico into Texas. No, that's not
likely,
true either.
not likely. I just think it is such a coincidence that there would be two independent outbreaks of this relatively rare, right?
Right. It's a relatively, but we have outbreaks every year of this, by the way. So think of this. You go to Iowa, or you're in Iowa, or you have to go to Iowa. You're traveling, and you stop at a restaurant, and you acquire cyclosporiasis. Okay? Mm-hmm. About a week later, you start suffering from it.
Yes.
But you're now someplace else, aren't you? And you don't even remember where you ate because it was on the road. It was like you were on the New Jersey Turnpike, or you're on Route 80, and you stopped with your family, and you had lunch, and you had some salad, or you had a hamburger with lettuce and tomato, right? Mm-hmm. And so now, depends on your sanitary proprietary behavior. These people that were involved in the Iowa outbreak could be the source of another outbreak somewhere else, unless they're treated. Because their feces contains the oocysts, and that requires several days to incubate outside. So that requires unsanitary behavior. Let's say campers, right? So you take your family camping, and the little kids start to develop diarrhea, and you take them into the woods, and they defecate near a public source of, let's say, drinking water or on a field. And the next thing you know, you've got contamination of that source. And so it's possible that the original outbreak in Iowa gave rise to the outbreak in Texas indirectly from those first cases that now relocated from Iowa back to Texas, and the next thing you know, you've got another outbreak.
Iowa's pretty far from Texas.
No, that's not so bad. Come on, Vince. I once drove around across the entire country.
Well, clearly we will
I'm sure
follow this
a lot of other people
until they sort it out.
But it's easy to see how this could be confusing, though, because of this.
So they might be connected, you're saying?
But indirectly. Yeah, if they had a way of... wait a minute. We have a proposal to our listeners. How could you link all these cases together in another way?
Well, you could get the cyclospora and sequence its genome and see if they're related. Can we do that nowadays?
Of course. That's a
Dickson,
piece of cake.
Really? You can sequence quickly, Dickson?
Oh, I can't sequence Dickson quickly, but I can sequence quickly, yes. So we could take the genomes of these outbreaks.
I know that. I'm just joking. Obviously, you could do it in a few hours.
So that you do that for viruses and
yeah.
bacteria and protozoans, and this is a protozoan, so you can do it.
Are they doing this, you think?
I have no idea.
How would you get samples to sequence from the feces of
Not
people?
a problem.
Tons of them, right?
Yeah, tons of it is right.
Well, the problem is that some of these people are well,
but you've got 600 cases, and the samples from these cases are probably still available through the hospitals.
All right, so we can hope that that's going on, and we're going to see an
MMWR
MMWR report someday in the next 6 months.
Genetic analysis of...
Yeah, that would be cool, wouldn't it?
Yeah, it would be. Because the biggest outbreak we've ever had of this related illness, which is cryptosporidium, was in Milwaukee in 1993. And that was all from the same source. And they proved it by genetic... Cryptosporidium. It's another Apicomplexity. And by the way, you wanted to talk about malaria? That's another Apicomplexity. So today is about Apicomplexes.
You know, the last time we talked about Apicomplexity was a long time ago.
It certainly
So you're going to need to redefine what... And I know that you're not against doing that, right?
No, of course not.
Apicomplexity.
A-P-I-C-O-M-P-L-E-X-A.
P-L-E.
See, Vincent is sitting here with the advantage of Google at his beckoning. Whereas I'm relying on a deteriorating memory.
You could have a laptop or a tablet too.
I could do it
yourself
to blame. That's right.
Were there any Apicomplexity on that T-shirt that I sent you a photograph of? You don't remember?
No, I don't remember if there were any.
That's all right. The Apicomplexity are a large group of protists, most of which possess a unique organelle called the apicoplast and an apical complex structure involved in penetrating a host cell.
So, Vincent, let's name the ones we've already talked about, shall we?
Toxo, I know, because John Boothroyd talks about the injection machine.
Spit and kiss.
Spit and kiss. Cyclo is one of them. Cyclosporiasis. Cryptosporidium. I don't know any others.
Yes, you do.
Malaria?
Yeah, that's the one.
Plasmodium.
That's the one.
Why isn't it on this list? Oh, I see.
It is on that list.
These are coccidian diseases. So what is a coccidian disease? Crypto, cyclo, toxo. There's another one here, isosporiasis. We never talked about that.
Isosporia is a big deal for poultry.
Why haven't we talked about? We could talk about animal.
Yeah, we've done some
parasites
animals. We've done some animals.
There are people who are concerned about animal welfare.
There are.
And we should talk about their parasites.
Okay.
But anyway, the coccidian parasites, what makes them coccidian? They're round?
They all live in the intestinal tract. They produce oocysts.
And then we have malaria, right? And
Yep.
Babesia is also a apicomplexan.
It is.
By the way, the name Apicomplexa is derived from two Latin words, apex meaning
The tip of.
and complexus meaning
It's not just a simple thing. It's got lots of things associated.
infolds.
Well, complexities.
You know.
Complexities.
So they have a organelle and a complex involved in getting into the host cell.
That's correct.
Well, that's great. That's kind of just coincidental that we pick these two topics.
Right.
All right, so we're finished with cyclosporiasis, right?
For the time being.
Did I say it?
You did.
correctly?
You hit it right on the nose.
We finished that for today.
Yep.
Was there one more thing I wanted to note? No, I think that's all.
Right.
And now we are going to follow, we're going to talk about avian malaria.
We are.
There is a reason that is not related to cyclosporiasis, right?
For talking about it, you mean?
Yeah.
Sure. I mean, it's a very interesting subject, but it's a crossover subject that we're going to talk about now because there are also another podcast that will occur later on today called
That's right.
This Week in Virology, which
Correct.
was our parent podcast.
What does that mean? It gave rise to the other?
It did, actually.
In a way, it did.
Spawned.
Dixon, you and I started that podcast.
We did, and we
together.
haven't finished it yet.
And Dixon, you still don't understand the power of podcasting.
What do you mean, Vincent? Of course I do. Of course I understand.
You think it's
the
a
power.
game. You think
not
it's
a game. No,
toy.
No. It's an information delivery system.
I love to bother you.
You certainly do.
So on TWiP today, we're going to talk about a story that broke this week, which is about a group of avian retroviruses called Reticulo Endotheliosis viruses.
Right.
And the story is a paper published in PLOS Biology, which basically says that these retroviruses are not uniquely avian. They actually originated in mammals 20 million years ago.
20? Oh, I didn't know.
So they were able to date the origin of these. And they were introduced into birds in the 1930s by a contaminated stock of malaria parasite. And when you came back yesterday from Australia, I told you this, and you said, oh, let's talk about
Sure.
avian malaria, because the malaria strain that was involved in that story. And I do recommend everyone to listen to TWiP today, because we're going to talk about it. And you're going to be on TWiP also, right?
I am. We're going
It
to talk
was
it
called
up.
Plasmodium lophurae.
Right.
And that's an avian
It
malaria,
is.
right? It was isolated from a bird in the Bronx Zoo.
Apparently.
So, where should we start? At the beginning?
Let's start with avian malaria.
I remember when we did the malaria episodes, and there were 2 or 3, and one of them was a story you tell, 1 million times 3 or something like that was the name of it.
1 times 3 million, yeah.
It was a very moving story.
It was a very sad story, too.
And
Still is. But
you
this
talked
is
about the canary in the coal mine?
No, I talked about the canary in India.
Let's start from the beginning.
So
Sir
let's-
Harold Ross?
Sir Ronald Ross.
Close.
But not close enough.
It only counts in horseshoes or in bocce, too.
And in hand grenades. They say hand grenades and whatever. So,
You're so morbid.
no. This is just an expression. It has nothing to do with my ebullient personality.
Okay, let's talk about avian malaria.
Sure. Let's talk about the origin of the discovery of malaria.
Why is avian... Was it discovered in birds?
Yeah. It was discovered... No, it was discovered first in people by Laveran.
Wait, malaria was... Human malaria was discovered in Laveran. By Laveran, what year?
1870s, 1860s.
But it had already been discovered in birds.
Well, the term malaria is a Roman word.
Malaria.
It means a bad air.
Are you making fun of Italians?
I am. See, you get under my skin, I got to get under yours in some way. So that's how you
skin.
No, you've got very thick skin. It's hard to get underneath your skin.
So avian malaria. All right, but the disease caused by these parasites was known in Roman times, right?
Sure, but not the
before.
origins. They didn't know where
Right,
it came
of
from.
course.
So they thought it was from bad air
Right.
because it came from the swamps.
Well, the whole concept of infectious diseases and microbes came from Pasteur and Koch many years later, right?
And many other people too.
After them. But Pasteur and Koch were the big guys initially.
They were. They certainly were.
And
So the point is that the bird malaria became a research tool to look into the biology of malaria in other animals which you couldn't readily access.
Mm-hmm.
So when Ross was a British medical officer
Ronald Ross,
in India,
right?
he first of all discovered that if you fed Anopheline mosquitoes on sick malaria victims, you could isolate a stage of that parasite in the gut of the mosquito. He discovered that first. He was the first one to make that connection. And he got his idea from Sir Patrick Manson. You know why? Because Sir Patrick Manson had already proven that filariasis was transmitted by mosquitoes. So Ross used that knowledge that he obtained from talking with Sir Patrick Manson to initiate experiments to prove that mosquitoes also transmit malaria. Ironically, at the end of their careers, and the Nobel Prizes had just started, Sir Ronald Ross actually got a Nobel Prize for discovering that mosquitoes transmit malaria.
Among who?
Birds in this case. But he had made the original connection, though, between Anopheline mosquitoes and human malaria by proving that Anopheline mosquitoes biting malaria victims developed stages in their gut tract that normal mosquitoes didn't have. So therefore, it must be the malaria parasite.
Did he discover the avian malaria?
He didn't discover it, but he used it. It was Plasmodium relictum.
Plasmodium relictum.
That was the name of the species of malaria that Sir Ronald Ross used in canaries.
Is that
in India to prove the
right.
Koch's postulates, basically. He went through the entire cycle.
Now, is Plasmodium, what's the name
Relictum.
of it? Relictum.
Relictum.
How do you spell that? We need to put those in the show notes.
Of course we do.
Plasmodium, hang on, dude, I have to type.
Hey.
P-L-A-S-D-I. How do you spell it now?
R-E-L-I-C-T-U-M.
Is the natural host the canary? No.
It's birds.
Where did he get it from?
India.
No. Which bird did he get it from?
Canaries.
Really?
Yeah.
What? How did he?
Canaries don't live in the Canary Islands, by the way.
That's not what I'm asking. Why?
Well, they were native to the area, so he was using...
Were they getting sick? Did he...
Yeah.
But how did he make the connection with human malaria?
Well, he began with human malaria and then went back to canaries to try to find the same thing in canaries, and he did.
So this is a naturally occurring plasmodium of canaries and other birds. What other kinds
and other
of birds?
And other birds.
Wild birds?
Many other kinds. Sure.
Can you inoculate ducks with this strain, and will it infect them? Chickens, turkeys?
Good question. I think it has a broad host range with regards to avian species. In fact, we'll get to that. Because
there's a great review article, by the way, that you have put in the show notes here, which is Ecology and Conservation Biology of Avian Malaria.
Right. And that's relictum, by the way. That is relictum. And relictum in Hawaii has caused havoc with the bird population.
There are other bird malarias,
but of course, there's a whole spectrum of malaria out there. And
P. relictum has one of the widest host ranges, occurring in more than 400 species and 70 different avian families. Whoa.
Yeah.
Then there's also Plasmodium elongatum, which is 67 species.
Then there's Gallinaceum,
Hermanni,
lophurae,
which we've lost.
Right.
It's gone.
You mean it fell off your radar screen, but there's a book by Barnum called...
P.T. Barnum?
You know, Vincent, you're so disrespectful of the old literature.
What's wrong with P.T. Barnum?
Plenty. Is P.T. Barnum. You're trying to make it like a circus of some sort.
The exit is there.
No, do you know where P.T. Barnum lived? He
No.
lived in Connecticut. There's a P.T. Barnum Museum in Connecticut. You can go visit it if you'd like, if you want to just clown around. But the point is that P.T. Barnum Was
No,
it
it's not
respected?
PT. His initials are not PT. I made
think
that
it
up.
is. No, I think it is.
Wow.
It's
I'm going to look it up. Go ahead.
So he wrote a book, a definitive book of his day called The Malarias.
Okay.
And it's a very, I actually had the privilege of meeting him once. Because Bob Gladz is a malariologist who studies mosquitoes, but his specialty is malaria. And at a meeting that Bob was at, and I was at too, Garnham actually was at the same meeting, and Bob introduced me to Garnham, and I was in awe of a man who dates back to the original discovery of mosquitoes transmitting malaria.
Yeah, his initial is, I can't find it. Garnum.
Garnham, Garnham, Garnham.
Google said, "Did you meet Gagnum malaria?" That's ridiculous.
No, Garnham. There's one called Garnami because it was named after him, of course.
So gallinaceum is
A species.
gallinaceous birds, right?
Yeah, that's right. And most of them are passerines. You know what that means?
No.
They can grab a hold of something with their claws and cling to them, like eagles, for instance, and
Those are
parakeets.
passerine birds.
Yeah,
right?
because they sit,
right.
as opposed to what kind of birds?
Gallinaceous,
No, web-feeded birds.
like ducks.
Yeah.
What do you call them?
Non-passerine.
Is that right?
No, they're called waterfowl.
Waterfowl. Okay.
I don't know the exact.
And these
term.
are very strict in the sense that they stay in birds. They don't go into mammals.
That's correct. And in fact, the actual species of malaria that transmits them, the anopheline
Species of
mosquitoes,
mosquito that transmit them.
anopheline mosquitoes do not transmit bird malaria.
I was going to ask, which mosquitoes? Culex?
No, well, I'll go back and I'll rearrange that statement.
Okay.
Only human malaria can be transmitted from human to human by anopheline mosquitoes. No other genera of mosquitoes can transmit malaria from person to person.
Okay.
But I'm not sure about my statement when I said that anopheline mosquitoes don't transmit bird malaria. I think they might.
Which mosquitoes transmit the bird?
Culex, mostly. Culex.
Mostly Culex?
And there's one in particular called quinquefasciatus.
Yeah.
And that's the one that's found in a lot of different places.
So mosquitoes like to bite birds.
They do. They feed on their blood all the time. And
How do they catch them? Birds are pretty fast.
they're passerines. They sit.
Oh, I see.
And then the mosquitoes.
Now, what did you tell me once?
they
They're
bite
sitting
them
in the
just around-
tree and they smell the CO2 coming out of the tree from the birds?
That's
Is that
correct.
right?
Yeah, that's the West Nile story. We're going to go back to another story, but
All right.
but Culex mosquitoes are important in that transmission cycle as well. And the way they detect the crows is at night when they all roost. And imagine how much CO2 is coming down out of a tree that has
Okay.
hundreds of crows in it.
Got it. And that's how the mosquitoes
And
find
mosquitoes
them.
find them.
So the birds at night typically roost.
Yeah,
They rest.
they do.
Are they like sleeping, basically?
Something like that.
And then they will stay there until the dawn and then go off.
And that's when these mosquitoes have a field day, so to speak.
Well, then
That's when
don't
they
the birds eat the mosquitoes as well?
Some do. In fact, bats are noted for their...
Bats are not birds, though.
That's true. But they look like birds.
All right. So do the birds get sick from having malarias?
They die from it.
How long does this take?
Well, it varies with the species. But with Plasmodium lophurae, when it's transmitted to ducks, I think the cycle is like 14 days, and then they're dead. And the remarkable part of that is if you have blood that you take from a dying duck, right, and inject it into a whole new series of ducks, it all develops at the same rate. So
Right.
at any one time, you can get Plasmodium collected from like all 10 ducks that you've infected, and they'll all be at the same stage of development, so you can accumulate a lot of one stage this way. That's why they worked with it.
Got it.
So Sir Ronald Ross, then just Ronald Ross, in India, worked with Plasmodium relictum
Okay.
and canaries. And canaries, if you go to India, you'll find that there's a lot of vendors selling lots of different things. And I think he was in Bangalore, and and there are a lot of vendors that would sell these little birds in cages. And
Right.
that's the source of his experimental models. But you'd have to say, well, that's not a very good experimental model because it's not genetically the same, and it's not the same age, and not the same sex. You know, like we go through our protocols now for our sciences. If you want to do an experiment, let's say, as I did with Trichinella, I would line up 3-month-old male mice of CFW strain and infect them to make sure that everything else was the same except for the one thing I was changing in the experiment.
Right.
With Ross, in those days, they didn't have those rigorous protocols to go by. But nonetheless, he still discovered something very important. And that was that the Culex mosquito, if it bit and took a blood meal from an infected canary, could transmit that infection to an uninfected canary.
Mm-hmm.
And that was the bridge. And by the way, Sir Patrick Manson tipped him off that that's what he should be working on.
How so?
Because he said, look, filariasis is transmitted by mosquitoes.
Right,
and so you're going to go to India, so why don't you look for that? And he did. He was helped along the way, all the way. So when Sir Patrick Manson and Sir Ronald Ross were considered for the Nobel Prize, they could only give it to 3 people.
Right.
So they elected to give it to all 3 people who worked on malaria. Sir Patrick Manson never worked on malaria. And yet he was the one that was responsible for the idea that led to the discovery.
Oh, well.
And we know lots of stories like that. Well, it just turns out that Sir Ronald Ross was an interesting personality. He was a gifted mathematician, a gifted musician, a gifted writer, a gifted poet. He even wrote a poem about his own death as he was dying. And there was one other thing that they listed.
Musician?
No, they listed that one. I think he was an amateur actor also or something like this. In other words, he was an expansive personality. And I think Sir Patrick Manson was of the opposite sensibility, let's just put it this way. So they became really at odds with each other in their later years.
Yeah.
Patrick Manson was really bent out of shape that he wasn't considered and given the Nobel Prize for the ideas that he...
Ideas are not enough, right?
No, they're not.
You have to reduce them to practice. You have to
You
get
do.
data.
But there was another group that was bent out of shape that they didn't get the Nobel Prize also for malaria. And that was a group in Italy that proved that Anopheline mosquitoes only carried human malaria. And they got their idea from Ross, but they were the ones that discovered it. But apparently Ross had already discovered that inadvertently because in India he had used Anopheline mosquitoes to examine the stomach contents to show that a malaria-infected patient can infect Anopheline mosquitoes and you can find stages of this parasite inside. So that was Ross's original observation.
Can we find avian malaria in the U.S.?
I believe we can.
Why don't we have human malaria?
We do have human malaria. It's all traveler-based.
Yeah, it's not indigenous, right?
No, but the last indigenous cases before the sporadic outbreaks that we've had since then was in 1951 in Minnesota. We used to have lots of malaria in this country, tons of it.
So
we got rid of it.
We get rid of it, yeah.
Because an expanding population needs an increasing food supply.
Right.
And so instead of maintaining those nice wetlands and swamps that we had throughout the lower Midwest,
right,
in places like Arkansas and Alabama and Mississippi and Georgia and places like that, what did they do? They filled them in.
I see.
And they grew
They
pigs.
got rid of the malaria, right?
Right.
Sorry, they got rid of the mosquitoes.
Right. It was an environmental control program, inadvertent.
Yeah.
But after a while, of course, it was vertent. That is to say, they did it on purpose. And they got rid of most of the malaria transmission zones this way, but not all of them.
In the summer in my town, my little town in New Jersey, they spray for mosquitoes.
They do.
Why do they do that?
They do that for eastern equine encephalitis there.
And West Nile?
And West Nile somewhat, but eastern equine.
What are they spraying with?
That's a good question. In your town, you should go find out. I think they're probably spraying with something like malathion.
And they always announce it down there.
I know.
I get an email.
Yeah.
Tonight we will be spraying.
That's right.
On such and such a street.
And does it work?
Who knows? I mean, the trucks only go down the street and spray, right?
So remember,
and the streets are a small fraction of the total land mass.
Of course.
Right?
But we still have mosquito control boards in New Jersey. So there are 21 mosquito control boards. And the best way to control mosquitoes is?
Get rid of the standing water.
Bingo.
Yes, but how do you do that?
But if you go back to, like, say, late April, early May of this year and track the rain events, we've had a number of rain events.
Yeah, we've had a lot of rain this year.
Rain, and has it been very hot? No, not really.
Not so hot.
So how about West Nile virus? Have we had much of that?
No, when we have
2
rain.
cases.
Yeah. When we have rain, the mosquitoes bite the birds that are hanging around.
The birds are still
Yeah,
around.
yeah.
Sure.
That's the Dickson hypothesis.
No, it's everybody's.
All right, so human, we can have human malaria in the U.S. You think we do
can.
have bird malaria?
I do think so.
But it's no consequence to humans.
That's right.
Why do we care about bird malaria?
Well,
Besides as an experimental model, which I fully understand, is it of any consequence to turkeys, chickens, farm types of birds?
it could be.
Does it
could
wipe
be.
out them? Does it wipe out?
No,
No?
they're susceptible to other kinds of infections. It's not a vector-borne
So
infection.
why do we care about avian malaria?
Well,
then?
if you're an ecologist and you are interested in the causes of extinction and population control,
yes.
then it's an extremely important area, because if you look at island communities, and particularly Hawaiian island communities, there are a bunch of bird species called bee eaters and honey. Let's see, what was the other name? Honey somethings. I'm blocking. I know the audience is laughing like crazy. They know the answer, but
What are you trying to say?
I'm trying to say that there are numerous genera of
Honeybees?
No, these are birds. They're all birds. They're called honeycomb eaters. No, they're not. We can look that up.
What do you want to know?
I want to know what the dominant genera of Hawaiian bird species are. The genera, not the species, just the genera. They're called, the common name is honey.
It's honey something?
Honey something.
The Hawaiian honey creeper.
Right! The honey creeper. No
They're passerine birds.
They certainly are. And they, in the old days, before invasion, right?
Invasion of what?
Before the whalers discovered Hawaii as a way station, all right? And not just a way station, it's where you're going to kill the whales, too.
Way stations between what and what?
Well, the Antarctic and the Arctic.
Okay.
They introduced Culex pipiens inadvertently. It's the dominant vector for lots of bird malarias. So the vector was there first,
right?
And then somehow a bird carrying bird malaria, in this case Plasmodium relictum, was introduced into Hawaii, infected the mosquitoes, and the mosquitoes then began transmitting it to these wild honey creepers. And 5 genera, not just 5 species, but 5 genera of honey creepers were extincted by a mass extinction event, which was traceable right back to the introduction of malaria into Hawaii. So you're asking what's the importance of studying bird malaria? That's the importance.
So in this article,
yes,
it says there are only a handful of documented Plasmodium deaths of wild birds in the U.S. So a handful. So this must be one of them.
Right. Because Hawaii is now in the U.S., but this happened before Hawaii was a state.
It's talking about it here, Hawaiian Islands.
Yeah.
Culex transmitted P. relictum has devastated native Hawaiian forest birds.
Exactly. Honey creepers.
But it says also there is relatively little evidence that avian plasmodium causes major epizootic die-offs in most natural bird hosts.
You know why? Because they're not island communities, that's why.
Yeah.
So here's the deal.
There's some zoo outbreaks that there.
Yeah, that's right. Honey creepers, by the way, if you go to Hawaii, have you ever been to Hawaii, Vince?
I have, sir.
Have you ever seen something on the history of Hawaii? And
probably.
the Polynesians that settled in Hawaii first.
I read Harold Michener's book. No, it's not his name. Harry Michener?
Yeah, you're right.
Is that historically accurate?
Harry Reasoner is the guy you're thinking of, probably.
James Michener.
James Michener. That's right. Hawaii.
It's in there.
It's an epic novel, right? It's
huge.
An epic. It's enormous.
Is it right?
Of course.
It's historically accurate. So it's a historical novel.
It is. So you've seen in the Hawaiian Museum in Oahu, there is an enormous collection of these ceremonial robes that were worn by the kings of these Polynesian tribes that settled there, right?
Right.
And do you remember what the robes were made out of?
No. No idea.
Feathers.
Okay.
Feathers of honey creepers.
Oh, these are the birds that got wiped out by the malaria.
Well, some of them, before the malaria arrived, were wiped out by the Polynesians.
Yeah, taking them for their feathers.
And if you go to Papua New Guinea, you can find that there are lots of birds of paradise that no longer exist, but the headdresses of the indigenous people that live there are decorated with the feathers of extinct birds. It's tragic because, we've inadvertently or even on purpose done these things to nature. So studying population dynamics from the perspective of parasite-controlled populations, it's a wonderful set of natural experiments that were conducted by nature, of course, but our introductions. Right. And it just shows you how powerful parasitism can be in controlling the numbers of things that are out there. Okay?
Well, that's what we've always said. That
So,
parasites are a way of controlling population.
So the point is that parasites are good for lots of things. They're not so good for looking at the way they affect human health, because obviously they detract from human health, right? But in this case, they teach us about the interrelationships of animals.
So you would imagine that climate would play a big role in this by controlling the mosquito population.
Yeah, that's right.
As well as the bird population.
Absolutely.
Geography, right?
You bet.
All sorts of environmental factors. So that's another interesting aspect of studying avian malaria. You could look at all these factors, right?
And you can look at it from this perspective of human malaria, too. Like, say, for instance, in Kenya, malaria was found at a certain level of altitude.
Right.
All right. And with global climate changes, those altitudes have now changed, and they're getting higher and higher because the planet is getting warmer. So
they mention in this article that there are lifelong reservoir hosts. What are the reservoir hosts for these avian species?
These are resistant birds.
So they're birds who are infected, but they don't get sick, and they live forever.
Right, and here's another thing.
I see. We should study their immunology, shouldn't we?
Well, wait a minute. There's an interesting study in Hawaii on honey creepers and the development of resistance against malaria in honey creepers.
Well, it makes sense, right?
Of course, population dynamics.
Yeah,
you select 90%, they all die, 10% are left, they breed, they replace.
Right.
Bingo. Next thing you know, the parasite is also changing a little bit to increase or decrease its pathogenicity.
Absolutely.
Due to passage, etc. So it's fun stuff, but it's also real stuff. I mean, that's how the world works.
Right.
And so parasitism at all levels, including viruses and fungi and bacteria and protozoans and helminths, they all have things to teach us.
It says here that on the island of Hawaii, emergent populations have been found in low elevation forests.
Right.
Also says that some efforts have been made to develop vaccines.
Why would you want to do that?
No, it doesn't seem like it would be something you'd want to do.
No.
But they do say that immunizing wild bird populations is a significant challenge. It's very
significant.
Maybe you'd want to release modified mosquitoes instead, right?
Look, I can imagine a very small island with an endemic species that you want to save, and there's no other way to do it except this way. It might work. But it's a segue into the next malaria story.
There's another story?
Oh, yeah. It's an exciting story.
Really?
Yeah. It involves human malaria and a recent clinical trial using a vaccine, a live parasite vaccine, Vince. When you say live virus vaccine, it raises the specter of, is a virus alive? And that's another total subject which we have fought over since we've started TWiP.
We didn't fight over it. We agree.
I meant that we have struggled with. How's that? I meant to say struggle.
No, we've discussed it a lot. It's a very interesting.
But in this case, it's real. It's real. So
I understand
what they've done,
that parasites are living.
What they've done is they've taken mosquitoes that are infected.
Who's they? Who's they?
The investigators. In this case, Stephen Hoffman.
Yeah. Okay. Go ahead.
Right. And they irradiated the mosquitoes to the point of destroying the ability of the parasites inside the mosquito to complete the life cycle.
All right. But the mosquitoes are still living.
And so are the parasites. They've just been attenuated. And in this case, I think they use cobalt, or radioactive cobalt, in order to irradiate the mosquitoes. Because mosquitoes, and by the way, insects in general, are very resistant to the effects of radiation. An enormous difference between mammalian species and invertebrates.
Okay.
So now what do you do? So now you've got attenuated sporozoites, which is the infectious stage for mammals, in this case for humans, right?
Got it.
What do you do with that? Well, Jerry Vanderberg at NYU, and we discussed this in a previous episode, also did the same thing with Plasmodium berghei and proved that irradiated sporozoites can induce 100% protection against a challenge infection from a mosquito that has normal sporozoites if you first expose them to these attenuated sporozoites. And that's what led the group at NYU to say, well, since there's this circumsporozoite antigen, it's the same antigen over the entire surface, that must be the immunogen that we can use to vaccinate against malaria. Well, that turned out not to be true.
Right.
Despite the fact that all these sporozoites from all these species share a very common theme. So there's something else there that's not detected on a live sporozoite, which is exposed during the active infection, which stimulates protection, which is permanent, in addition to this circumsporozoite antigen. So Jerry Vandenberg is given credit, at least I give him credit because I know him and I know he did this work first. That work was picked up again by Stephen Hoffman's group as an act of desperation.
Yes.
Because no molecular vaccine has ever worked against malaria. None. They've tried and tried and tried. It's like the AIDS story with HIV antigens. It's the same story for a different organism. So now here we have the opportunity to go back to the original observation, taking Plasmodium falciparum infected Anopheles stevensii, irradiating them so that you attenuate the sporozoites, and initiate an infection which is mosquito-borne, or taking those sporozoites and injecting them to see what happens to those volunteers. And so here's what they did. They did these experiments about 2 years ago, thinking we've got Vandenberg's experiments reproduced, and we should get 100% protection, right? They only got 20% protection. Well, how come? And the answer turns out to be the route of distribution of these sporozoites. If you inject them into the skin, like you would assume mosquitoes are doing, then you only get about 20% protection. But if you inject them intravenously, then you get, in this case, this is a brand new finding, you get in one group that received about 130,000 sporozoites, they were completely protected against infection. I think it was 12 volunteers. That's a miracle. Okay? That's a brand new finding that just came out like 2 weeks ago.
Where?
Good question. So where did I read this?
We have to put the link in the show notes.
I know. So if you type out on Google Stephen Hoffman malaria live attenuated malaria vaccine, you'll probably get the reference for it. I just read it. I think it was in Science, by the way. So we now have the proof that not only did it work in mice, but it can now work in people as well. So then...
I see here.
So
August 9th.
That's right. So Stephen Hoffman...
Unconventional vaccine shows promise against malaria.
Unconventional. Well, it's the oldest way of producing a vaccine, basically. Right? Because the oldest vaccine that worked is...
The oldest vaccine against what?
Doesn't matter. I can tell you what it's against, but you wouldn't know that one.
Smallpox.
Well, that's one of them. You use scarification and you're using attenuated virus.
Yeah,
but there's another one with leishmaniasis that's even older than that and uses a scarification for that too, and that works as well. Those are attenuated leishmania organisms. So here we have a series of attenuated live vaccines. If you want to consider vaccine as a vaccine, a live virus.
Okay.
But it's at least infectious. How's that?
Right.
Leave out the word live. Just say infectious. All right? And that clears up the issue. Is it alive or not? Who cares? It's infectious. It's attenuated, but it's still infectious. So here we have, you can add malaria to that list now. That's a very short list, Vince.
The title of the paper is Protection Against Malaria by Intravenous Immunization with a Non-Replicating Sporozoite Vaccine. Dickson, intravenous is a very difficult way to deliver a vaccine.
No, it isn't. Oh, you mean for people? Oh, it's an incredibly difficult way.
So this was done in human adult.
That's right.
9 and 6, very few, less than 20 subjects.
Correct. But...
And they, I presume, used subjects in an area at risk for malaria?
No.
No?
No, of course not.
What did they do?
In Rockville, Maryland. Then they injected them they allowed mosquitoes to feed on them that had the malaria.
Well, look at this. 5 of 6 non-vaccinated controls developed malaria following controlled human infection.
Correct.
Oh, my gosh.
It has to be a strain of malaria that's easily cured.
And 0 of 6 subjects
0 of 6
receiving 5 doses developed malaria following infection.
So the point is, it's proof of concept. This is a proof of
I'm
concept.
amazed that you can give people malaria, Dickson.
Why not?
They're going to be sick the rest of their lives, aren't
They
they?
are not. Of course not.
They get over it?
They're given a drug. The moment they detect a parasite in the blood, they give them the drug.
And that's it?
That's it.
And these people are fine the rest of their
In
lives?
fact, they may be even better than most other people because now they're resistant to malaria.
And they're using falciparum.
They are.
All right, so why is the thing in that kids get repeatedly infected? They get malaria every year, as you've said many times.
But the degree of infection decreases over time. So finally,
Why doesn't natural infection protect them?
it does.
They simply get reinfected
But
because
by the
there are
time
a lot of infected
8
mosquitoes.
infections, every season you get infected for 8 years, you lose something that's almost intangible to measure, and that is your ability to reason.
It doesn't make any sense, Dickson.
What do you mean it doesn't make any sense?
You want to develop a malaria vaccine, yet natural infection does not confer protection.
That's you've misstated it, Vincent. You're misstating this again. Let's go
I
back to the
understand
beginning.
It modifies the disease, but you
No.
still get sick.
No, you're
Right?
not listening. You're not listening again. I'm going to have to repeat this again. Let's start again.
Okay, natural malaria.
You're a little kid.
Yes.
All right. Let's say you survive from the age of 8 months to 5 years old. Why is that such a window of opportunity for malaria to kill you? Why isn't 0 to 8 months?
Because you got maternal protection, right?
Thank you.
All right.
So that's one level of protection.
All right, so then beyond that, you get...
Number 2, there's this window in which the maternal antibodies are low,
Right.
and you could die from an infection because your immune system isn't quite ready yet to take care of everything, right? But after the age of 5 years old, the mortality rate is almost 0.
Okay.
Now, why is that? Because you can fight off the infection before it kills you. Okay.
But you're sick, right?
You get sick to begin with. That is infection number 1. Infection
You
number
recover.
2, you get sick. Infection
Right.
number 3, you get sick. Infection number 4, you get not so sick. Infection number 5, you're maybe not so sick. And maybe infection number 10, by the time you're 20 years old, you're not getting very sick anymore from malaria. You might be sick for a couple of days, and then you can just go back to work again. Now that's protection against the lethality. That's what they're interested in, Vincent. They're not interested in whether you get sick or not. They don't want you to die.
It was always my impression that to make these individuals have productive lives, we have to make them well, because malaria makes them sick enough so that they can't do anything.
It
right?
does. So what if a vaccine allowed them to acquire malaria that only made them sick for 2 days?
That would
That's-
not be bad.
That's great, as a matter of fact. That would be welcomed by open arms.
I presume that this would prevent lethality.
Yeah, that's what they're trying to do.
So second, third, fourth, and fifth infections in people, are they ever lethal?
Sometimes, but
Not as much as the first infection.
That's right.
All right.
Yeah. I mean, if you get infected between 8 months and 5 years of age, you're probably going to die.
Now, the other issue with this vaccine is that they have to dissect the salivary glands
It's
to
maddening.
get the sporozoites.
It's maddening, Vincent. It's maddening.
That's terrible.
Well, you know what? There's another way around it, of course, but the other way around it may not work, and that is to just let these irradiated mosquitoes feed on people.
Right.
But so the reason why I'm touting this at all is that this is the very first time ever that any preparation for malaria other than an infection can induce total refraction of the infection. This is number
Yeah,
one study. Okay, so now you have to build on this study. What are the steps necessary to take forward this finding and turn it into a viable vaccine?
Wow, the guy who did the study says that they have to do a bigger clinical trial,
Of course, number one is proof concept.
and they have to figure out how to make sporozoites faster.
Here.
So right now, they have a team of 12 to 15 dissectors,
Yeah, I
and
know.
each of them can tease 150 mosquitoes an hour,
Right.
and they need to increase that.
So, what was your subject that you worked on, Vincent,
When?
when you were a graduate student?
Influenza virus,
Okay, as a postdoc.
polio virus.
Thank you. And so, what was the breakthrough in polio vaccines? There was a breakthrough, Vincent. I
Cell
know
culture.
that. Yes. And who got the Nobel Prize for that?
Anders Weller and John Enders, Weller, and
Somebody's medium.
wait a minute.
Eagle.
No, it wasn't Eagle.
I thought Eagle got it too.
Enders. Oh, this is terrible. Wellams.
Yeah, Weller. Weller.
Enders, Robbins, and Weller.
Robin.
Enders, Weller, and Robbins. Gee, I'm just getting old.
No, you're not. You're getting better. You're getting better at this.
Anyway,
So I'm leading you to something.
The breakthrough for polio
was tissue culture.
Tissue culture. So we need a breakthrough for mosquitoes.
You
right?
need a tissue culture for sporozoites.
Oh, well, that's not so easy, Dickson.
Wait a minute.
In the meantime, people are dying, so they're going to dissect the mosquitoes.
It wasn't easy for polio either. That was very difficult work. In fact, I knew Dr. Weller. I knew his son. His son is still active.
They have to develop a culture for sporozoites.
Right, and
So,
that's
Dickson,
the breakthrough.
If you were going to do this
Not do it. Why don't you rephrase that? If I were Bill Gates Jr.
Yes.
and I wanted to invest my money wisely, I would invest it in insect tissue culture work. I would invest billions of dollars, offer huge prizes to people to switch their fields and get involved in insect tissue culture to culture specifically the sporozoite of Plasmodium falciparum.
Falciparum.
It's falciparum. So
I think that's a call. I hope Bill is listening.
He might not be listening, but if he is, or other people are listening, they could suggest it to him. So what you're going to end up with, right, is a race for the prize for the person or people or groups that develop an effective sporozoite culture system for malaria. Now, we already have an in vitro culture system for the erythrocytic phases of malaria. And you were going to ask me something about that today, too. So I will just tell you that the person who worked on Plasmodium lophurae and probably is responsible for distributing this virus, the reticulo endothelial affecting virus.
Reticulo endotheliosis virus.
Yeah, and what is reticulo endotheliosis, though? What's that disease?
I don't know, Dickson.
Yes, you do. It's an infection of the reticulo endothelium system.
I guess.
So what is the reticulo endothelium system?
It's the system in our blood that filters.
It's your immune system.
Parasites.
It's your immune system.
Part of it. It's part of the immune system.
You've got Kupffer cells and T cells and B cells. That's all part of the reticulo endothelial system.
Reticulo endothelial system.
Yes, because reticulo
macrophages. It's an old term, actually.
I know that, but it's stuck.
For the immune system, the part of the immune system that consists of phagocytic cells, which are located in reticular connective tissue, primarily monocytes and macrophages, and they accumulate in lymph nodes and spleen.
And liver. Don't forget the liver, because it's got a Kupffer cell.
The Kupffer cells of the liver.
So what if we could train Kupffer cells to intercept sporozoites on their way to the liver? That would do it.
Why? Why would you do that?
Because that's where the infection starts in malaria, in the liver. That's why I'll do it.
So this is something different now.
What we really need, so what we really need is an effective tissue culture system for the mosquito stages of the parasite. Because that's the definitive host.
Yeah, I'm sure people have been working on that for years.
Yeah, right? But not at billion-dollar investments.
Interesting.
Yeah, very interesting, I think.
So these people got 135,000 sporozoites each.
Yeah, 5 times.
So these mosquitoes are raised in the laboratory, so they're clean, right?
Yeah. You mean of other viruses?
Yeah. Dickson says he doesn't know.
I hope they've looked into that, because if they haven't...
How would you look into it?
Oh, it's very easy to do, actually.
How?
You do PCR.
You could PCR for the viruses you know, but maybe there are viruses you don't know, the unknown knowns.
We have the entire...
Unknown unknowns.
No, we have the entire sequence of Anopheles stevensii.
Yeah, I think you would have to deep sequence this strain of mosquitoes to make sure there's nothing else in it.
You got that right, and I'm sure they did that, because
I'm not sure that they did it.
These are good molecular biologists.
I am not sure that they did it, Dickson.
Yeah, so the other approach, of course, is to improve the molecular vaccine by finding out what is it in a irradiated sporozoite that exposes itself to the host that allows it to develop full protection. Right. What is that other missing antigen or antigens? And we don't know. We don't know. But it wouldn't be a bad thing if we discovered how to grow sporozoites to confluence ad libitum in like a spinner culture, for instance, right? And irradiate them and use those as vaccines. What would be the biggest objection from the public, do you think, Vince?
To
get the vaccine. What is the vaccine? It's an irradiated sporozoid.
Right, why is that a problem?
Irradiated. Don't you know that radiation, you say that, well, we irradiated these, and now you're going to be given them. People will think that they're getting radiated.
Well, it's just...
I know.
There's no more radiation left.
No, I know, but they don't know that, okay?
Well, we have to educate them then.
Oh, good luck on that one. So that might be a stumbling block. It wouldn't be the stumbling block for the Army, because I think that's what they want to do this in. They want the Army to have a vaccine for malaria, because it's a big problem no matter where you go in the tropics. And, of course, travelers would love to have a vaccine that they could take rather than mefloquine and all those other nightmare-inducing drugs that they have to take once a week in order to prevent disease but not infection, remember?
Right. Cool.
So malaria is still very much with us and very much on the tips of the tongues of those responsible for funding malarial research. In fact, if you go to NIH grant giving in the tropical medicine section, 90% of the money is given for research on malaria.
I would not be surprised if Gates Foundation is involved in culture, as you suggested already.
Well, Stephen Hoffman didn't rely on any of that money to do this study.
This is a U.S. government-funded study.
Right. He used to work for the Office of Naval Research.
And you know Stephen Hoffman.
Very well. No, very well. He's a good friend, and I haven't seen him in a while, but he and I have spent time together over the years, and perhaps we should have him on a TWiP. Call him up, get him on, and find out his take on all this. I'm sure he's going to be excited about it. He's going to be inundated with press now.
So he's at NIH?
No, he's a private foundation, private company.
Which is Sanaria.
Sanaria, yeah, and
Sanaria.
He's a co-founder of that company with his wife, who studies angiogenesis. Well,
cool.
She did.
Now, what does this have to do with avian malaria? Nothing.
No, it's just another malaria story.
It is a malaria story. So now it's hard to title this episode.
Oh, it's a potpourri.
No, it's not. What do we call it?
It's a parasite grab bag. Do we have any email to read?
Yeah, we do.
Good.
We have lots of email.
Should we?
Yeah, for sure.
Oh, good.
Tons.
Right. Are there any more parasite stories that you're aware of, Vincent?
Oh, there are lots, but that's enough for today.
Yeah, nothing in the news as far as I'm concerned, except that there is one more.
Well, there's the 1P lophurae story, but we're going to talk about that on TWiP.
There's another parasite story
in which case you're going to tell us all about Bill Traeger, right?
I would like to, yeah. I knew him very well.
Okay, but there's another parasite story?
There is. There's the first human case of anaplasmosis.
Really? What is anaplasmosis?
That's a good question. I don't think I used the right term. I think that's a bacteria.
Yeah, it's anaplasmosis is a tick-borne disease caused by a bacterium.
Yeah, that's not the right word I was looking for. There's a protozoan parasite. It's related to Babesia. Tick-borne, tell you what, type out tick-borne protozoa.
Protozoa.
Tick-borne protozoa. I bet it shows up.
Tick-borne protozoa.
Right? Anything?
No, nothing shows up.
Tick-borne parasites.
Tick-borne parasite.
Someone out there is laughing. Says, I would do my whole work on this. How come these guys don't know this?
Oh, that's 2011. Now, you're still talking about something that just happened here.
Yeah. But it's
so
known.
Here's a list of tick-borne diseases in the U.S.
Okay,
so we have anaplasmosis, babesiosis, ehrlichiosis, Lyme disease, Powassan, which is a virus, rickettsia, Rocky Mountain spotted fever, southern tick-associated rash illness, tick-borne relapsing fever, tularemia, and 364D rickettsiosis.
No idea.
This is a new disease that has been found in California.
Okay, so anaplasma is not ehrlichiosis because they listed those as two separate things.
Rickettsia.
Didn't they?
Sorry.
It's a protozoan.
What is?
It's a protozoan infection of animals.
What is?
The one that was recently reported as the first human case. And I'm trying to think of the name of the parasite.
Is it tick-borne, though?
Yeah. And I can't remember the name of it.
Well, you can look it up, and we'll do it next time. How's that?
Okay, that's my homework assignment.
Let's do some email.
Or we could make it a contest and
Well,
give
we
away...
did that a few episodes ago. No one answered.
That's
so
too bad.
No more contests.
All right, fine.
Alan writes, you have the best podcast on the web, hands down. I use your TWiP episodes in both my undergraduate and graduate classes.
Wow.
Thanks.
Oh, you're welcome. Where does he teach?
University of the Nations.
And where is that?
I don't know. He writes, I've recently, you could look it up, but you don't have a computer in front of you. So
but you
instead
do.
of reading the email, I have to look up where the University of Nations, and it's in Hawaii.
Okay, great.
And he writes, I've recently seen a family in Hawaii with recurrent pathogenic blastocystis hominis and Dientamoeba fragilis.
Right.
And I'm suspicious if they are getting it from close association with feral pigs. I see a little published on the possibility from Italy, but do you know how prevalent it is in pigs? And particularly, does D. fragilis even have a transmissible cyst stage?
Does not.
Are both of these parasites considered emerging pathogenic diseases?
Well, we talked about the ambiguities associated with proving anything to be pathogenic because of the numbers of cases of positive infections, but low clinical cases numbers. So we have huge numbers of positive infections with very, very low numbers of people suffering from a disease that you might want to attribute to that pathogenic process. So the answer is we still don't know, and of course you can't complete Cook's postulates in this one because of the ethicalities involved. Particularly because you can't get rid of it once you've got it.
Are these considered emerging infections?
No, blastocystis is not emerging. In fact, it's been around a long, long time. So,
How about
Dientamoeba
Dientamoeba?
fragilis? You know, as a technician, I worked tirelessly through a hot sweating microscope all day long, and it was rare. It was very, very rare occurrence to find Dientamoeba fragilis. Very, very rare.
Any suggestions on setting up an investigation to answer these questions?
Well, I would, yeah, sure. I mean, the one thing you could do is connect up with some veterinarians
Yes.
and look at the pigs first and find out what the prevalence in pigs are of either one of these protozoans, and then try to make some epidemiological connections between the amount of contact to pigs and the prevalence of these infections also in people. That's one approach, of course, but then you're still stuck with whether it's actually the case or not that they did catch them from the pigs. And, you know, there are a couple of parasites in pigs that you can catch as well. So hard to do it. It's very difficult to do it.
The next letter is from Andrew, who writes, Hi there, my name is Andy. I was wondering if you may be able to provide some feedback on something I'm developing.
Uh-oh.
I'll try to make it as explicit as possible. First off, I was wondering how difficult it is to engineer a virus's ligands to match up with a desired host cell's surface receptor. The relevance behind my first question is, would it be possible to engineer a virus that can attach to T. gondii surface receptors and ultimately infect the parasite? Of course, there are lots of things to consider, but I'm really only curious of the concept itself. I haven't found any literature that specifically applies to the question, so I've turned to you. Any thoughts? If engineering is possible, viable infection would be the first step, and then developing an idea on how to kill the parasite would be the following, in my amateur perspective at least.
Well, there is one already that infects Trichomonas that you might be able to model the Toxoplasma gondii episodes on. But
Are there any T. gondii viruses?
I don't know of any.
So you can easily modify viruses to bind to different cell surface molecules, but you have to make sure that those are internalized and allow the virus to get into cells.
Yeah, I mean, the rest of the process is very specific for the virus.
And then once the virus is in, it has to be able to replicate.
Right.
Or if you're just going to deliver, say, a lethal gene, it may not have to replicate, but just deliver it.
But you might not need a virus then.
Well, a virus is a good way to deliver things specifically. And, for example, adenoviruses can be modified to target them to different kinds of cells.
Can't you do the same thing with liposomes?
Well, then it's not specific, right? If you're
No,
using the receptor to make it specific.
Well,
But in this case, he wants to hit Gondii specifically.
Can you
right?
engineer a liposome to have a specific receptor?
You could.
That's what I think.
You could.
Because I know cancer drugs are being delivered this way somehow.
So you can, the answer is you can engineer viruses. But the problem is, since we don't know of any viruses of T. gondii,
Right.
it could be difficult. But I guess if you could have it bind a protein which would be internalized and then release the contents of the endosome, that might work. Do T. gondii do endocytosis? They're not cells. They're multicellular, right?
No.
Unicellular.
Protozoans.
Well, they could take up things.
They have to take up things in order to feed. Plasmodium is a better target here because their food vacuole is actually inside the red cell, and they have a huge amount of hemoglobin that they take up at once, and then they digest it and sequester the heme portion. So, that you don't need a receptor for that. You have to get it through the red cell membrane, but once it's there, you might be able to engineer some kind of a drug that would kill off the plasmodium that way. And I think, lots of drugs already get through the red cell membranes anyway.
Next one is from Carl, who writes, "Hi, you guys. I'm Carl from Northern Ireland. I would like to thank you for all the weird and wonderful information about parasites, viruses, and bacteria, and general information. It has been a pleasure writing about it, so I don't forget it, which keeps me informed and can read over to myself all the parasites. I'm a chef in the food industry.
Oh, marvelous.
Which reminds me to be careful when handling raw food products. I always wear latex gloves to keep me protected, and think that more people in the catering industry should be put through a parasite awareness course for themselves.
I'd love to give that
others.
course. I would die to give that course. That would be great.
I say this because of my own experience of having a parasite in my body, which thankfully was taken care of. I am now a vegetarian and have never felt better. I have evolved at last. Ha ha. It's like the Adam and Eve story and the serpent, and to choose what to eat and knowing what's good for us and what's bad for us. The ancients spoke and drew out the Ouroboros. You know what the Ouroboros? Now I understand what it all means.
I don't.
Now, we are part of the food chain, not at the top. So it makes me think, what is the purpose to be food for parasites? So they can live and survive? Makes me wonder if Mars has water.
Mars does have water.
Maybe there are parasites that exist there also. Anyway, don't write a book about it. Just thought saying thanks again, and look forward to continuing to listen to both of your podcasts and many more interesting conversations, as not many people want to talk about parasites.
Oh, nice.
Ouroboros. Let me Google that, okay, Dickson, because we have to learn things.
I don't know what that is.
Oh, it's an ancient symbol depicting a serpent or dragon eating its own tail.
Ah.
You've seen that, right?
I guess.
A serpent eating its tail. That's what an Ouroboros. He says now he knows what this means. There's a cycle. Stephen writes, by way of an introduction, I am a retired IT professional who took an undergraduate degree in biology many years ago. TWiP, TWiV, and TWIM are wonderful listening, and I particularly enjoy the informal format of the broadcast. Dickson, I am a person with celiac, and I cannot help but wonder if celiac evolved and survived because it might have had a survival advantage for ridding the body of parasites, which lodge in the small intestine and are hidden from the immune system. Ongoing regular treatment by gluten would not be great for health and longevity, but perhaps symptomatic or seasonal treatment by gluten-containing grains would confer some added benefit for primitive man. Can you please speculate on this possibility? Thanks.
Because you get diarrhea from getting, and that kicks out the parasites. No, that doesn't work like that. In fact, parasites induce diarrhea, so that probably is not the process by which you'd like to eliminate the parasites. What you really want to do is stimulate your immune system. So in celiac and in malabsorption syndrome cases and in people with Crohn's disease, they have actually done geohelminth therapy by giving them trichuris or hookworm, and it has alleviated the symptoms from those conditions, metabolic conditions that are present, and probably you have a genetic basis for that. And very few people have Crohn's disease. More people have celiac, I think. So,
Okay.
a few worms won't hurt you. A lot of worms will. That's the hypothesis, and in fact, it's got some validity in clinical medicine. So I would follow that line of reasoning.
Thanks, and keep up the great shows. I always look forward to listening to them on my not an iPod during my daily dog walk. Kevin writes, after all the recent fishy episodes of TWiP, I thought you folks would appreciate this GIF circulating under the title Trout of Nowhere. So an animated GIF, Dickson. This is pretty funny. It's a trout jumping out of the water, and the guy catches it on the boat. Do you see that?
Right, that's very funny. That's very funny.
It could happen, right?
This is a tourist that looks like more like a salmon than a. But that you know where that is? I bet you that's in New Zealand, because they have these jet boats in New Zealand. That looks like a New Zealand river. That could be a very large trout that jumped into the boat.
It's pretty funny,
That's
huh?
a riot. That's a riot.
Would you have thrown it back?
Of course. And I will tell you also
It's
that...
a big fish. It's probably a salmon, right?
No, it could be a trout in
Could
New
be.
Zealand. But that could, they have some salmon in New Zealand also, by the way. This is really, really off the subject, but there's a place in Mexico called Ixcalac. It's on the Yucatan Peninsula, almost into Belize. As a matter of fact, it's only about a couple of miles from the Belize border.
Mm-hmm.
And this is a place where you can go and try to catch a variety of sports fish. And one of them is the tarpon. Tarpon is a valued sports fish, and it's not a food fish. You just catch it and throw it back. And tarpon live in mangroves. And so this guide that we were fishing with that day took us to a backwater of the lake that's really an estuary of the ocean to find some tarpon. And on his way through these mangroves, he scared about a 40-inch tarpon, and the damn thing jumped into the boat.
Nice.
And he picked it up and posed with it as though he had caught it, and then he threw it back in the water. So maybe I'll try to find that picture and send it to you.
All right. Want to do a few more, Dickson?
If we have time.
We have time. We have to
Sure.
stop soon and get ready for TWiP. But
This is
we
all
can
true.
do a couple more.
Well, let's do two more.
All right. Next one is from Dorotha. I am a huge fan of your show, and I really appreciate the work that you do to make science more approachable. I especially like TWiP because I think that parasitology is very underappreciated, not because it's any less exciting than other disciplines, but possibly because of the yuck reaction many people have when hearing the word parasite. Your show is just another proof that parasitology can be interesting in a non-horrid way. I guess you can apply Terence words, homo sum humani nihil a me alienem puto. I'm sure Dickson will enjoy translating it, as I noticed he is really into Latin. She says you can apply that to parasites. And that's a famous quotation by Terence, by the way, which means, I am a human being. I consider nothing that is human alien to me.
I would agree with that entirely.
He was a playwright of the Roman Republic.
I would
by
have
the way.
guessed that.
You would have guessed it, huh?
Just, off the top of my head.
I have a question for you that came to mind when eating my garden-grown plums, which turned out to be more organic than I expected, with some worms enjoying their taste probably as much as I do. Are there any parasites that use worms or, in general, insects as vectors to get into their host via oral route? Thanks for the great show.
The answer is yes. There are. But if you ask me to name them right
But
now.
to get into us?
Human infections, no. This is mostly animal infections, mostly
Okay.
of veterinary importance, bovines, etc.
So they can use worms. The worms will bring in the parasite?
Paratenic. They're called paratenic hosts.
Right.
They don't actually develop any stages inside. What they do is that they ingest something, and then you ingest that, and then you end up with the other thing. Yeah, there are some examples of that, sure. I think in parasitology, since it's such a broad field, whatever question you ask about transmission cycles, the answer is yes. It doesn't matter what the question is, because there are so many varieties of ways of getting parasites. That,
All right. So we have two more because they both deal with the same subject.
Okay. Well,
Andrew writes, you've probably already been notified on this, but the Coriolis effect has nothing to do with the way water spirals out of a drain.
Right.
On a body of water the size of a sink or even the Oculus of Singapore, the Coriolis effect is far too small to have a significant effect.
Correct. I would
The
agree.
direction that water swirls in the toilet is determined by the shape of the water jets around the rim of the toilet. It gives a link to the Coriolis. And then there's an email from Greg, who writes, Dear TWiP hosts, concerning your discussion near the beginning of TWiP number 57, which is the Coriolis effect. As a youngster, I was fortunate to witness a demonstration of the Coriolis effect right at the equator in Kenya. My family and I watched a man drain water out of a plastic bowl with a hole at the bottom. A few meters north of the equator, a matchstick floated in the water, spun clockwise. Literally a few meters to the south, the experiment was repeated and spun the other way. To my amazement, directly at the equator, the water drained straight out without any rotation.
Oh, brother. Now
Only much later, as a student of physics, did I learn that we had been deceived.
Of course.
The apparent acceleration caused by the Coriolis effect is of the order of the velocity of a moving object times the angular velocity of the Earth. Even if we generously give the draining water a velocity of a meter per second, this gives an acceleration that is 100,000 times smaller than the acceleration due to gravity.
Sure.
So other effects will easily determine which way water drains out of a bowl or a toilet bowl for this matter. I speculate that the man doing the demonstrations at the equator, quote unquote, would shake the bowl slightly, perhaps even unconsciously, to get the desired outcome. The Coriolis force has a noticeable effect in phenomena occurring over longer timescales, accumulating the effect of the very small acceleration, such as weather patterns or the precession of a Foucault pendulum, or in phenomenon with very large velocities, such as the trajectories of missiles.
Wow.
I hope you will excuse this digression into the world of classical mechanics and its applications to detecting fraud in the tourism industry. Keep up the excellent work, Greg. I think it's great that people of different interests listen. And
fantastic.
This is why, Dickson, in closing, you really need to buy into this new media thing.
Vincent is convinced that I haven't done that. Of course I'm buying into it. I love this.
But you always travel instead of twipping.
Hey.
You should not travel.
Oh, really?
I know you like to travel, but don't do it.
I think if you talk to people in parasitic disease research, they've always traveled.
Okay.
That's the way you learn about these things.
And now you're going to be gone for another 2 months, right?
I will?
That's what you told me.
Where the heck am I going?
Who knows? You're always coming up with something.
No, I'm not going anywhere.
TWiP is a podcast available on iTunes and microbeworld.org/TWiP. If you like what we do, go over to iTunes and leave a rating or a little review. It helps to keep us visible and get more people to learn about parasites, which I think is very important. It's not yucky at all, right?
No,
Dickson?
not at all.
Please send us your questions and comments to twip@twiv.tv. My host here on TWiP, Dickson Despommier, can be found at verticalfarm.com, trichinella.org, or trichinella.com. Which one is it? And medicalecology.org.
Yes.
2 .orgs and 1 .com.
Correct.
Thank you, Dickson.
You're welcome. Pleasure.
Anything else?
No, I think it's great.
Did you eat lunch yet?
No.
I guess you're hungry, right?
A little bit.
All right. I'm Vincent Racaniello, and you can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.